yeah, it's very exciting to talk about the future of bioimaging. I think what we're seeing now coming along is the conjunction of new technologies, both in terms of hardware, but also probes and new physics that are used for inspecting living systems, together with computation, advanced computation, machine learning, deep learning, and other technologies such as large language models and large foundational models that are all converging towards a very fascinating future of possibilities. Not just in life sciences, frankly, but I think in all scientific disciplines, really. And I will try to distill some facets of our work that align with this theme of the future of bioimaging, and I hope you will enjoy these stories. Just to give you a bit of context, I'm a group leader at the Zuckerberg BioHub. That's a private non-profit research institute in the Bay Area. We really are a hub between three universities, UCSF, Berkeley, and Stanford. And there I lead a team that tries to combine these three disciplines together: optics, computation, and biology. And the biology we're interested in is developmental biology. So we're trying to understand how embryos develop. And to do that, the bet I made when I started my team was to try to combine all these technologies, both on the hardware but also on the computation side, to try to answer the fundamental question of how an organism develops. And as an aside, I'm currently hiring, so we have two positions on the optics side, but also soon two positions on the computation side, possibly. And so if you are interested, or so if you know someone, let them know, and then you can always find my email in my paper, so you can track me down. So the things that we've been developing span all the way from ways to rear our fish in our fish facility, automating everything from the life support system of the fish, but also automatic feeding of the fish, building instruments, which is something we will talk about today. Together with Juan Iglesias and others, I was part of the initial team that started napari, and this has been an incredible success that has grown thanks to a fantastic community. By necessity, we've had to develop algorithms for terabyte scale image processing on the data sets obtained from our microscopes, simply because you have to somehow come to terms with the size of the data and the challenges to analyze it and just handle these pixels, just process them. On the analysis side of things, I will talk to you today about how we attack the problem of cell tracking, which is a key issue when we try to understand a developing embryo. And I won't talk much about it, but we've been also working on ways to improve the quality of our images with tools for denoising them, and trying to make these things very practical and easy for people to use. And we have also lots of side projects currently. This is one of them that involves self-supervised deep learning in the context of collaborations with other group leaders at the BioHub. But really, for us, really seeing is the first step towards discovery. So I will start with the instruments that we are designing. So for those that don't know, light chain microscopy is a form of microscopy that is very, very special, because we essentially illuminate our sample from two sides with a very thin sheet, and a thin sheet of light. And because our samples are fluorescent, and the structures of interest within our samples are fluorescent, we can detect, we can image that illuminated plane with another objective. And this allows us to combine these images together into 3D images. And we can do that very fast. And because you only illuminate what you look at, and you only look at what you illuminate, this is not only a fast way of imaging samples in 3D, but also it's very gentle. We don't kill the samples. Typically, they swim after they've been imaged for hours. And this is actually volumetric. So we get 3D images typically every 30 seconds. We can get a 3D image. And so we have time lapses that describe the whole process, the whole arc of development throughout time. So, and if you look back, at one of the branches of development of this technology, one of the ever since back in 2004, 2008, people like Yusken and Keller worked on the very first light chain microscopes for fluorescence samples, we had these two objectives, one illumination objective and one detection objective. And then one branch of that has been to add more objectives around the sample, more illumination objectives, more detection objectives, to the limit, and then people have realized, oh, we can use the same objective for illumination and detection. And this is, something that's typically called a DEXP microscope. And then even more recently, the team of Philip Keller has and Raghav Shetty and his team have developed this completely crazy microscope that has 4 illumination, 4 detection, and they're all the same objectives. Amazing technology, very complex to build and operate. In my lab, I continued some of the work I did in Philip's team, and we basically built, when I started the lab, a multi-view light chain microscope with 2 detection arms, and here 2 illumination arms. Sorry, I'm gonna have to switch to laser. So these are illumination arms here, and these are detection arms in this direction here. And it's a beautiful instrument. It can produce beautiful images of developing embryos. For example, here you have a Drosophila embryo developing, 3 different views of that data set. So this machine that we have, inherited some of the technology I developed during my postdoc, which is to be able to adaptively adjust the parameters of the microscope as the imaging is running. So this microscope is adaptive, it's kind of a self-driving microscope. It's one of the first grandpa smart microscopes, if you want. You see here correction curves that describe all the changes that the microscope is operating on the system to try to keep things aligned. It's a great machine. We've been using it for zebrafish imaging. So here, is a view of a 2-channel recording on our light chain microscope showing, here in cyan all the nuclei of all the cells, and in purple here you have one type of cell, the mesoderm, that's lighting up. And this is really a beautiful data set that's been quite popular on Twitter. And it was acquired by Merlin and Jordao, and has been doing some processing on that to produce these beautiful images. So how do we mount the sample in this system? We put the sample within a chamber that has these 4 objectives surrounding it. And we have to really, for each sample, we have to create a whole universe of hardware parts around it to be able to make it happy, keep it alive, and then in place within this rather complex system, right? It's not a very ergonomic system. This is a microscope designed for physicists more so than for biologists, but it works. But then, there are some issues with it, right? So one of the issues is when you have a multi-objective system, you have a lot of objectives around the sample, and biologists would prefer to have their sample there versus having so many things around it. So that creates a limited space. It gets complicated to have, horizontal surfaces on which you can mount the sample, which is easier than to have the sample in a gel within a tube. This is really complicating our imaging protocols. So can we do better? Well, it turns out that you can. It turns out, use a single objective to both create the light sheet and detect that illuminated plane. And this is exciting because now you can imagine, mounting the sample on a horizontal surface and using gravity, which is a very practical feature of our universe, or at least of our, planet. So you can just have a piece of glass and have the sample on it, and you can have multiple wells, you can have multiple sample. Instead of having just one sample, you can mount multiple of these samples together in arrayed in such a manner. You can also imagine, perturbing the system from above, adding drugs. So this makes the instrument a much more biologically useful instrument. So zooming into this a little bit, the idea is therefore, to have a single objective that, produces a light sheet, which is typically a very thin sheet of light, and then detecting the cone of light emanating from that plane, from all the fluor forces within that plane. And this is something that, became possible once, since the seminal work of Dunsby in 2008, which showed that this was actually possible. And there's been a flurry of activity since then where people have been perfecting this approach. And I think one of the most significant advancements was the work of Bin Yang, who was at the time not in my team, but in the team of Bo Huang at UCSF. And he showed something that was remarkable, is that you can actually achieve this oblique illumination and detection without compromising resolution. He showed that all the light, all the light emanating from this secondary objective, and I will explain a little bit more what that means, can be collected, and therefore, you can have this very odd oblique illumination and still preserve high resolution imaging. And in this case, for cells on the dish. The problem is, of course, we're not interested in cells, at least in my team. So we are interested in embryos, and the embryo is much bigger than a few cells on the dish. So how do we achieve a much larger scale imaging without compromising resolution, speed, and all, and in particularly the ergonomics provided by the single objective setup? So this is the work we've done in this direction, and it was published last year in Nature Methods. And really the work of Bean and a large team of folks in my team, but also in other teams within the BioHub and some collaborators. So the microscope is called DaXi, meaning that it's a microscope that can image large things at very fine resolution. This is how it looks like. Unfortunately, it's a little pixelated. It's so big, the screen, that you see the pixels. But it's a rather complex system, but it's also in many ways a very straightforward instrument. So let me show you the list of key features that this instrument has. It has a single objective in direct proximity to the sample. There are other objectives hiding behind it, but they are not in proximity to the sample. It's capable of high resolution and large field of view imaging, or large volume imaging in more general terms. It can scan very fast and very long. And we have a dual illumination system. So we have a bit of that ability to image the system from different sides, and therefore acquiring different views that we can combine together into a better image. So let's look at the single objective aspect of this work. So in a traditional light sheet system, you have an illumination objective that will create a sheet of light through your sample, and you have another objective, a detection objective, that will look at whatever is emanating from that plane. If you put a tube lens, as you should do as whenever you build a microscope, you basically recreate the distribution of light here. And one interesting property is, because of the magnification, you have a squared magnification along the axial direction. So your sample is actually deformed. And typically, what people do is they put the camera there, and they really and essentially what you're doing is trying to acquire on the sensor of the camera the distribution of light over that plane. But the truth is that if you move that camera, you would image other parts of the sample, right? If you have an oblique illumination, that doesn't work anymore because, well, it works, but then you have this very inconvenient configuration where the light sheet is, because of this elongation along the z-axis, this angle here is very steep. So if you try to put a detector there, that's never going to work, because you would have grazing angles. The light will bounce, will reflect off the sensor. That's never going to work. If anything, if you could do the opposite and compress the sample on the z-axis, that could work. But that's not something we can do with optics here. So instead, what we do is we use an almost identical objective, and there's some variations to that theme. Then and you can recreate an image, a virtual image of the sample that is from anything that you put after this. It would look as if the sample is there. So this is the closest thing to transporting an object. It's not there physically, but its light appears as so if it was there. So what you can do now is you can put now a new microscope with an objective, a tube lens, and a camera that will then image this at an angle. The beautiful thing is that you wouldn't be able to do this here because you have this objective already here, right? You cannot put two objectives superimposing one on top of the other. But here the sample is not really there. The geometry here is very amenable to this, and so we can now image this here. And the beautiful thing is, if these two objectives are identical, then you have very nice properties in terms of the quality of the image, and you can explore the design space a little further than that. And there's a very beautiful theory developed by Butcherby in 2007, and further refined in the work by Dunsby, and you can understand how these things work. And it's quite a powerful process. It's called remote focusing. We can recreate the sample there, and original terminology of remote focusing comes from the fact that you can change the focus by imaging at different planes this virtual image here with this secondary microscope, if you wish. So in the end, we use these ideas to build a microscope where the samples here and some glass surface on the stage. We have a primary objective, we have a relay, we have a galvo that allows us to scan the light sheet through the sample. We have a system that we'll explain later that can flip the orientation of the light sheet. Eventually, we need to decouple the illumination light that comes, this is the blue line here, from the detection light that eventually goes to this secondary and tertiary objectives. And here we use, I will explain that, we use a very special tertiary objective that has very special properties. But this is roughly the overall layout of the system. And so with this system, we can now, because the sample is conveniently on a glass surface, we can have arrayed imaging where we have high throughput. It's high throughput from the standpoint of light sheet microscopy, perhaps not from the standpoint of classical high throughput cell imaging, but 9 embryos at a time. Of course, we have too much multiplex imaging, right? So the temporal resolution suffers. But we could always have a smart system that would eventually figure out which one of these embryos to further image, for example. This is really possible because we have removed a lot of optical complexity away from the sample. It's still there, and in some case, in the movement of that complexity itself creates more complexity, but it serves the purpose of the biology. So another feature is that we actually kind of retain high resolution, high imaging volume in such a system that is amenable for easy imaging. So the way it works is that what we typically do is we scan the sample through the light sheet. And as I explained, that light distribution gets teletransported, if you wish, into the space between the secondary and tertiary objective. And so what's really happening here is you have this virtual sample scanning through the surface of that very special objective called SNAUT version 2. This objective is very special. And just to say here, we retain high resolution, almost diffraction limited. And this system has an NA of 1.0, meaning it has a high resolution, much higher than the 0.8 NA resolution that we can achieve with our multi-view system that I showed you earlier. And as I mentioned several times, there's this special objective that was designed by Alfred Militsekin and Andy York at Calico. They designed the first version for cellular imaging, and the new version is capable of imaging much larger samples. And here it's very special because it has a working distance of 0, an NA of 1.0, so high resolution, and a large field of view of 450 microns. Why the working distance is 0, and what can that work with? Because we're imaging a sample that's not really there physically. We're just imaging the light distribution that was moved away from the sample. So it's also important because we actually can capture through the refraction through refraction caused by this piece of glass. We can that is in direct proximity to the plane that we image. We can capture all the light rays and therefore preserve resolution in our system. And this allows us to image entire larvae and keep things over one dimension. At least we have almost an infinite imaging volume. Of course, you have to trade-off speed with the size of what you're imaging, right? And if you look at it, it's a very nicely you can see almost all the nuclei over a very large volume. And if you inspect this data set, it's a beautiful. Here, this is a zebrafish larvae, by the way. We can recover beautifully different structures throughout the anatomy of the fish. So that's great, but are we still fast? And why can we scan so long and retain resolution? So so the scan range, you get a sense of it. We're scanning a sample through the light sheet, and you could scan as much as you want there. But initially, we were thinking of scanning the light sheet and not move the sample. The sample here is this box, in an abstract way. And that's limited in scanning range. But if you move the sample, there's no more limits, right? Because a stage can move, that supports the sample can move, millimeters. The problem is that you would have motion blur. So here what we do is we move the light sheet in such a way as to cancel the movement of the sample while we take images. So when we expose, nothing moves. The sample does not move relative to the light sheet, nor relative to the detection plane. But when we move the light sheet relative to the sample, that's when the camera sends the information back to the computer. So this trick makes it possible to go as fast as a galvo in terms of scanning and as far as a stage. And that allows you, as I explained before, to image very large volumes up to a depth of 300 microns. Along this dimension is pretty much unlimited. And along this axis here, it's about 800 microns. So plenty of space to image developing zebrafish embryos. And this is a beautiful example that we did with, in collaboration with Tom Kornberg at UCSF, where we image, Drosophila fly oocytes over, 3 millimeters of tissue at very high speed. I think it's in order of 30 seconds per time point or so. I mean, you can see it here. Every half a minute you have a new image, and there's more pixels here than I can visualize on a 4K monitor after projection. So another key aspect of this microscope is the multi-view. So we added why do we need multi-views? The thing is that when you send the light sheets through the sample and you collect light, turns out that, well, the collection light goes to the same cone of, same cone, but the light has to go through different parts of the tissue. So it experiences different obstacles. And so for every point in the sample, it's likely that there's going to be one image that's going to be better. So the idea here is that we require images for each one of these. So we require 2 stacks with light sheets on one direction and that light sheet in the other direction. And then we combine that together. And so this is really why we need to do this, is because you need to bring the light in different ways in the sample, and ideally also bring the light out of the sample from different directions. And so here you would expect that one image would be better than the other. So to be able to do that, we have this trick where we have a flipper system with a set of 2 galvos. We flip the number of mirrors that the light experiences, and if you remember, your high school geometry. You can essentially change the reflection by the number of mirrors that you hit. So here's a bit of a schematic of this. So in one configuration, the light path goes through only one reflection here, through these fixed mirrors, or 2 reflections. And if you do that, you actually have essentially flipped one half of the microscope relative to the other half. So it's as if I was literally flipping this part relative to this part. But we don't do it by physically moving things, but by flipping the light itself. And this holds for both illumination and detection. And that's the beautiful thing, is here all the optics are common to both illumination and detection. So whatever you do to one, you do to the other 2. Things stay focused, everything is the same. And now what happens is you acquire overlapping volumes corresponding to two different views, and therefore 2 different light sheets at different angles. And you basically now have the possibility to combine this information to get better images. So if you look at these 3 locations here, one view will be definitely better than the other. So you want to use more information from this view than from this view. And in other locations, because of the local geometry of the sample, it's going to be a different story. It's going to be the other view that's better. And so for every pixel in the image, you need to choose which of the 2 views you need to collect information from. And we have algorithms that can by estimating the information content in these images, combine these images in ways that would maximize the information content. Sometimes, frankly, it's just better to pick one versus the other locally over some region than to try to combine the good with the bad. Here, for example, this fusion here, perhaps is, probably just keep this and really never consider information of this image. And this is something we tend to do these days. When you do all this and you acquire a time lapse of a developing embryo, you get these beautiful videos of of a zebrafish embryo developing. You have, here the tail bud, which is a tail fish. Here the eyes are somewhere here hiding. You can see a lot of cell divisions, a lot of, this, what you see, all little dots here are nuclei, right? The nuclei of cells. You don't see the cells themselves. You see the nuclei, which are proxy for the cells. And here are zoomed in views of single planes to that. And you can appreciate how complex that developmental process is. And we need to see better and more to be able to understand this. And this helps us definitely get there. We wrote a lot of code to do the image processing and image analysis of this. Joe Dow in particular has been maintaining this repo DEXP, and then he has now a new version of it, which is DEXP 2. And this is what you use to to process our images and then make these videos. That might be of interest to some. Okay, so that was the instrument part. But we cannot just stick to just images. We have to go beyond. We have to actually get from these raw images to insights, to knowledge, possibly, right? So we developed a lot of software and computational methods to turn images into insights, all the way from tracking cells in entire embryos, using LLMs and things like ChatGPT to try to do image processing, image analysis for us, using self-supervision to either decode the protein localization code or denoise images in a user-friendly way. And napari is one thing, again, the project which we started with Juan Inés Iglesias, Nick Sofreneff, and there's like quite a few members of the community here today. But I won't talk about this part because I already talked about napari this morning, and there's been already a workshop about it this morning. But and I won't talk about the self-supervision here because there's no time really. So instead, I picked two projects that are very, very recent. One doesn't really use deep learning, but it illustrates the fact that as we move towards the future, we should not forget the past, and sometimes deep learning is not always needed. But of course, if you would add deep learning to this, perhaps things would get even better. And another project is really, a very forward-looking project where how to use ChatGPT to do image analysis for you. So let's start with cell tracking. It's really the work of Joe Dow in the team. And it's really a program that aims to track cells in 2D and 3D and in multicolor over small data sets, but also very, very large data sets. So the features of this software are that we can use segmentation hypothesis from different segmentation algorithms and parameters. So we can the starting point is if you have your favorite segmentation algorithm, you can use that, or you can combine the results of different segmentation algorithms to get better results. The output of tracking is not just the tracks themselves, but the segments that are best suited for the tracking. It supports, as I mentioned, 2D, 3D, multicolor, large, small data sets, and it's easy to use. We have lots of notebook-based examples, and it supports a variety of data formats. And really, the starting point here is that segmentation is hard, and mostly because tracking is hard because segmentation is hard. Right? So if you look at this, I think this is actually, I'm not sure if it's Cellpose or Stardist, but it's hard. It's sometimes typically what you have to do is, for your particular data set, you might have to tune things a little bit. So it's very difficult to, segmentation has progressed dramatically, and I think, Martin here and others have been, at the forefront of that. But it's difficult to have that silver bullet that will always perfectly do everything that you want. And the key problem that we have is that when you have a nucleus, for example, a nuclei of a cell, sometimes the algorithm get confused, and so it either thinks that this is a whole segment, or maybe there are two segments, and therefore maybe these are actually two cells. Are these two cells? Are this one cell? And we have typically to choose, and then once we have chosen, when we do tracking. And if we make a mistake here, it's too late, right? You can't possibly do tracking well if you make a mistake during segmentation. So the question is, do we really need to pick segments before tracking? Now, do we really need to commit to the segments before you do the tracking? So what if we could consider multiple segmentation hypotheses while we track? So one thing to understand is that to understand a bit the way we do things, is there's an equivalence relationship between segments and contours. When you segment an image, you identify these regions that correspond to the nuclei, and this is equivalent to contours that delineate these regions, right? If you run a segmentation algorithm, this is what you get. But we are actually interested in this thing. Why? Because there's a thing called multi-level contours or ultrametric contours, which actually represent not just a single segmentation, but a hierarchy of candidate segmentations. So you can imagine that, this hierarchy represents all sorts of possible interpretations of the image. Here, if you decide to cut the hierarchy here, that means that these are all the segments you're considering. But you could, consider other parts of the hierarchy and therefore find that, oh, actually, some of the segments should be merged. At the limit, you could say, well, no, actually, there's only two segments. This is one segment and this is another one. And then the multi-level contours, because the contours themselves have different gray level values, can represent this hierarchy and therefore this diversity of hypotheses in terms of what the segments should or are. Should be or are, yeah. So the limit, you can just have all the nuclei as one segment, which is definitely not the right answer, right? And in some cases, different parts of the image require different settings and different thresholds. And so it's difficult. You have to somehow, make decisions that are varying in space and time. And who is going to make these decisions, right? So we use this diversity of hypotheses during tracking, but how do we get them? Where do we get them from? So if you look at this example data set, if you apply, for example, cell pose here, you will have a hard time segmenting everything. So one thing you can do is play with gamma, which is a nonlinear transformation on the image. And you'll see, oh, because some cells are brighter than others. And so playing with some parameter here will help you a little bit. But it's difficult to find one setting that will solve it for everything, for every place and every time. So it's really, it's difficult to have one algorithm or one parameter value or set of parameters that can segment all cells at all places and all times. That's very hard. So what we do instead is we just segment. In this case, in this example, we have one algorithm, but we vary the parameter. And we get lots of different candidates of segments for this varying gamma value. And you can see here the differences. Sometimes, some segments appear and then they disappear. For example, this segment here is absent. This cell is not segmented, but it's segmented before. And then maybe, there's some other examples where, something is segmented here correctly and not there. So, every value has, strengths and weaknesses for different parts of the image. What we can do is we can combine all this into an ultrametric contour that will contain all this information about all these different candidates. And when we run our tracker on this, we get a segmentation which happens to be better. So essentially, the whole tracking on this data set is better if we combine all the segmentation together versus trying to find the best parameter for the gamma value. I think this is very important, as I'm super excited about this, because what it means is we can eliminate parameters from the segmentation algorithm. We can literally eliminate the parameters. This, you do not need to have a ground truth here because we put everything. There's no choice to be made. There's no decision to be made. You put everything together, and we know that the result will be better than even if you had guessed the best parameter for the segmentation. So you can imagine doing that over multiple algorithms, over multiple parameters per algorithm. And so I think this is very exciting because the parameters of magic numbers, magic values of parameters are really like the thing that breaks most algorithms, right? Because someone has to figure out what the parameter is. So how do we select the best segments? And this is where time plays a role, because you can have candidates, for your segments, and you can have these multiple candidates, but it's only time that will tell you that the segment is relevant. Here is an example of two cells that seem to fuse into one and then to separate again. This is what happens. Sometimes the images are poor and you can't tell the cells apart. So here what we do is we use time to figure out that, these, cells do not merge, at least in our data here. And so this must be 2 cells. And since we have 2 candidates here for this, we have the candidates. We know that it's possible to interpret this image as 2 cells. And so we use that because it's the only thing that makes sense when you consider time. And so then, we build, build a whole graph connecting time. The different, we represent all these candidates as trees, and we connect the trees by how likely is a segment to be the same across time. We have measures of how similar the segments are over time. And what we're trying to do is find the best interpretation, the best selection of segments that maximizes some sort of score, and at the same time respects constraints that are, for example, if you pick this segment, you cannot pick this segment because the segments you pick cannot overlap. Cells cannot overlap. So if you pick this segment, for example, here, select in blue, you cannot pick these two. So if you pick these two here, these 2 segments, you cannot pick this one, right? Because the choice of a segment of the identification of a cell will prevent other choices. And how do we solve this puzzle? We built, an integer linear program. I won't go into the details. It's rather complicated. It incorporates all sorts of, prior knowledge about the biology of the system, like, existence of divisions, cells that appear, that disappear, and has also the objective that is to make sure that we can connect the time points across time. It's complicated, but it works. And let's look at it in action. So this is a data set, a zebrafish embryo we're developing. And first you see, the head here, this is the tail. You have a lot of cell divisions everywhere. And eventually what we're interested in is to actually look at the segmentation. So, here's one segmentation, but we have all this, candidate segmentations as part of the data set, right? We can't visualize that so easily here. And once you have that, you can look at the tracks that are obtained by the tracking. So here you will, Jodo has visualized, the tracks. And so this is an early version of it. So there's quite a lot of mistakes still, but it's a beautiful video, so I keep using it. And then, if you look carefully, you'll see, there's cell divisions happening and so on and so forth. And the scale of it, so there's a lot of engineering that goes into making sure that this actually runs in a reasonable amount of time. Because the theory is one thing, but the implementation has to be performant. We have in order of 20 million segments across time that have to be connected across all these, hundreds of time points. We can look at a single cell. We can look at the story of a single cell through this data set, but I have a better video, so I'm gonna jump that a little bit. So, but here, okay, so here's one cell, we follow it, but the truth is I actually have a better video, so I'm gonna show you that. And that's the video where we follow one cell and we have the X, Y, and Z X slices that show you the complexity of tracking that single cell in this very crowded environment. And there are all the limitations that light chain microscopes have, which is like any microscope, poor resolution along the axial direction. And so that single cell is tracked, and it's one of many tracks that are followed here. And you'll see them appear suddenly here. So we have all that information, of course, right? And we can follow the story of a single cell through a cell division too. So here is again showing the scale of the problem. This is a region with one cell. This is a zoom of that cell in this very crowded environment. We need to make sure we follow it accurately. Eventually, that cell will do the thing that cells do, which is to divide. And then we follow the 2 daughter cells to their respective histories. And do mistakes happen? Yes. But I will show you that the results we have are pretty good. You'll see we have quantifications of the quality of these drugs. Yes, how accurate it is. So there's this cell tracking challenge competition, which has been running for 10 years. And Jordao made one submission, and he managed to get first place on 2 rather challenging data sets. Embryo scale, cell tracking in Cell elegans and Drosophila, On this data set, he has 4th place, but we hope we can get to the top eventually. But it's quite an achievement from Jordao, because this competition has been running for some time, and so and he has quite a significant gap with the 2nd place contestants. So I think we for our purpose we cannot trust it blindly, but we are confident that we have something that's very useful for our biology, right? And this is very easy to use. Just a few lines of code. He put a lot of work to make sure it's easy for others to use. And so you can get you can go to our GitLab repo and look at all the examples we have. We have Jupyter notebooks so that you can use it for 2D, 3D, and multicolor data sets and whatnot. And play all these tricks with these multiple hypotheses of segmentations, and see how well you can track your data sets with that. So I will come to the final piece, which is a bit more of a fun one, which is Omega. So Omega is a project that is really about making a napari plugin for ChatGPT, and it really started as a piece for Nature Methods that was published earlier this year, where Rita Streck, the editor of one of the editors at Nature Methods, asked me to muse about the future of bioimaging and what it holds. And 2 ideas came out of it. Like, we probably need better instruments and better probes, of course. But then very quickly in the piece, I go into this idea that maybe the future of bioimaging is really this idea of a dialogue between mind and machine. And what that means is that with the advent of large foundational models, you can imagine a future where you will have a dialogue with a computer, where you will present an image and suggest analysis that can be done. The machine will provide back annotations, segmentations, and will give feedback on that, either textual or via interaction. And through a dialogue that could dramatically accelerate, bioimage analysis and processing, perhaps PhD projects that used to last for three years would probably be done in an afternoon, perhaps, hopefully. And so as I was writing this piece, I was also monitoring a little bit on Twitter what people were doing with the OpenAI API in Python, and I realized that actually maybe you can do some of this stuff already. And so I started furiously started working on this project where I combine napari with ChatGPT, and it's really a way to have a conversation with the machine to do image processing and image analysis. So here are examples. You can interact with the napari viewer, asking Omega in a natural language to write code that will do things to the viewer. For example, changing the view, the dimensionality of the view, translating, rotating. You interact with natural language, and the system, through the magic of ChatGPT and LLMs, figures out how to write code that will do that to the viewer. It knows, ChatGPT knows about napari, knows about Python, knows about image processing, image analysis, knows about all the libraries that we know. It knows more about all this stuff than I would ever dream of knowing. It does not know everything, but it knows an awful lot. Very often what I ask works. And sometimes it misunderstands what you ask, and sometimes it comes up with some weird answers. But to a large extent, it's remarkable that we can already do this with these first chatbots that have emerged. And we are just at the beginning, right? Here's another example. I load an image in napari, and I ask to segment the cell nuclei present in the selected layer. It chooses to use studies from Martin and Uwe, has a way to write the code for this. And then I say, count the number of cells that are segmented, and it will count them and give me the answer of 317. He knows how to write code to do that. He knows how to comment that stuff. Beautiful, you can learn from it. You can learn how to write Python code to do all these things. Then I ask, write a CSV file on the desktop that lists all the segmented nuclei together with their area coordinates, sort the rows by decreasing area, and open the file when done. I didn't teach that thing to do any of this. It knows how to do this on the computer. And it opens the file on the desktop. And it, very nicely, explains to you how to open a file on the system, how to write and read CSV files and so on. Another example is to segment nuclei on the selected 2D image, which is this image, the same one, using studies, so the same thing. But now we tried something different. We ask Omega to make a widget that takes a label layer and returns a new labels layer, but filtered. Only labels within a provided area, range of areas, min and max, are kept in this new layer. So I'm asking Omega to create a little widget that I can use not just once, but multiple times. A widget is really a little interface with all its parameters. It literally knows how to write all this code. It knows about napari, and we never taught ChatGPT how to use napari. And now you can basically filter segments in a segmented image based on area. And you can test it, and it just works. And sometimes maybe it will make a mistake, but you can see the code, you can check it. You know, in the future, I'd like to be able to edit some of this code and be able to save some of the widgets. That would be great. Another example, make a widget that draws a scale bar in white on the lower right corner of a 2D monochrome image. Parameters should be length of the bar and pixel resolution in microns. And it will make an attempt at it. And I'm saying attempt for a reason. So it's slow. Some of these videos have been sped up a little bit. And it writes the widget, but there's a problem. It doesn't really understand about the fact that, you know, this is a gray level image, and so if you draw a bar of color 1, that's very dark. So I argue with the thing a little bit and say, you know what, that's not working. Please add me a parameter so that I can choose the intensity of the scale bar. It's happily obliging, and then creating a new widget that will have that additional feature, which is here, the intensity. And now it works. And now I can, you know, add my scale bar. This is one of my favorite examples, because the first time I tried it, it worked. I mean, when I recorded it, it made a mistake the first time. But the first time I tried it was an RGB image, and there was no issue with RGB images. Final example, make a step-by-step plan to segment nuclei in a 2D image. The nuclei are brighter than the background. So we're not asking an action, we're asking to think about how we go about doing it. It comes up with a very nice multi-step plan, and I can say, you know, I don't care about plan step 1 and 2. Go to step 2 and apply it. It remembers what it has told. You know, you all have used ChatGPT. You know that there's memory associated to this. And when you use the API, you actually have to implement the memory yourself. And then you can go on, proceed to the next steps, and make some variations on these steps, and say, oh, you know, let's do 2 variations. So you can, you know, you can modulate. You really have a conversation with something that's hopefully smart. And you can, you know, you refine things and then, you know, go backwards and reapply steps. And yeah, so, and you know, ask, okay, you just revisit the plan, you know, to make changes. Let's do step 3 with 1 opening operation. So, you know, you go through the thing, you explore a little bit, and then you basically can, you know, analyze and process and analyze images. And in this case, you know, the goal was to kind of segment images, right? So it's not necessarily, you can have this, you know, open-ended discussions and processes. Yeah, thanks.